In this video, I will be explaining the process of oxidative phosphorylation and the role of the electron transport chain. As always, I recommend drawing along yourself on a piece of paper so that you have a copy of the notes and you understand the content better. Oxidative phosphorylation occurs on the inner membrane of mitochondria. So I'm going to start off by drawing, first of all, the outer membrane of a mitochondrion. And then I'm going to draw some of the proteins that are in the inner membrane, starting with ATP synthase and the proteins that make up the electron transport chain. Between these, I'm going to show the inner membrane of the mitochondria. And you can see I'm not particularly exaggerating those folds, those cristae, but they would be much more dramatic in order to increase the surface area of the inner membrane. So we have our cristae, the folds of the inner membrane, the intermembrane space, which is the space between the outer membrane and the inner membrane. Outside the mitochondria we have the cytoplasm, and within the middle of the mitochondria we have the matrix. So in the previous videos we discussed the process of glycolysis, which happens in the cytoplasm, followed by the link reaction and Krebs cycle that happen in the matrix. The products of each of those reactions, one of the products of each of those reactions was NADH, the coenzyme high energy electron carrier. Krebs cycle also produced the high energy electron carrier FADH2, or reduced FAD. These high energy electron carriers are going to provide the energy needed to drive oxidative phosphorylation. This process starts when NADH drops off its electrons at the first protein in the electron transport chain. So the NADH is being oxidized and this first protein is being reduced. The NADH is oxidized to NAD+, which will return to glycolysis, the link reaction, and Krebs cycle in order to keep those reactions going. This is actually why fermentation is not needed when there is oxygen present, because this process is enabled to continue releasing NAD+. The high energy electrons that are now in the electron transport chain move down the electron transport chain through a series of protein carriers. The electrons from FADH2 also enter into the electron transport chain a little further down, oxidizing the reduced FADH2 back into FAD+ that can return to the matrix to Krebs cycle. In each step of the electron transport chain, as the electrons move down, the energy from those electrons is used to pump hydrogen ions, which have also been dropped off by the NADH and FADH2. It's used to pump those into the intermembrane space. So hydrogen ions from the matrix are pumped into the intermembrane space. This is going to lower the pH of the intermembrane space. So far this should look very familiar if you've done photosynthesis already. It's a very similar process. electrons with a lot of energy moving down a protein chain, causing hydrogen ions to be pumped across a membrane against their concentration gradient in a form of active transport. This buildup of hydrogen ions in the intermembrane space is going to lower the pH here. Be careful, some people get confused and think that this is hydrogen ions being pumped out of the mitochondria. They're not being pumped into the cytoplasm, just into the intermembrane space. That causes a concentration gradient between the matrix and the intermembrane space. Hydrogen ions will then move through the ATP synthase enzymes in the process called chemiosmosis. Hydrogen ions will move through ATP synthase in a form of facilitated diffusion as they're going through this protein channel. And as they move through, down their concentration gradient into the matrix, that causes the ATP synthase to spin. Just like in photosynthesis, the spinning of the ATP synthase provides the energy to convert ADP and PI, phosphate ions, into the energy carrier ATP. Something to note here, a lot of ATP is made in this process. Lots and lots of ATP. Estimates vary per glucose molecule, but you'll see numbers anywhere from 28 to 38 molecules of ATP as a theoretical number that is made by each glucose molecule. That's a lot of ATP, a lot being produced here compared to Krebs cycle and glycolysis. Most of the energy that came from those processes has been transferred to the electron transport chain. This process here, the flow through, is chemiosmosis, and this overall process is oxidative phosphorylation. Now you may realize that something is missing. What happens to these electrons when they get to the end of the electron transport chain? Well, these electrons react with two hydrogen ions. So we have two electrons, two hydrogen ions, and half a molecule of oxygen to form water, H2O. This water is one of the products of respiration. Remember, per glucose, 6 molecules of water are produced per glucose. You may have missed it, but this is why you need oxygen. When you were born, probably the first thing you realized, other than it's cold out here, is that you need to breathe. Later in your life, you may have realized, I need to breathe, that someone's told you I need to breathe because I need to get oxygen out of the air. And in GCSE biology or in earlier studies, you may have known that oxygen was important for respiration. Well, here it is. This is its job. What? Oxygen is the final electron acceptor. And this is critical to sustain life. You need the oxygen to pick up these electrons and form water. So why is it so deadly if we can't breathe? If we can't breathe and get enough oxygen to our mitochondria, the oxygen won't pick up the electrons. There'll be no oxygen present. If electrons are not picked up at the end of the electron transport chain, then there will be a build-up in the electron transport chain, and electrons will no longer be able to flow down it. That means there will be no concentration gradient of H+ ions, as none will be actively transported into the intermembrane space. As a result, chemiosmosis will stop, ATP synthase will no longer spin, and oxidative phosphorylation, the production of all that ATP, will cease. You can still produce some ATP from glycolysis using fermentation and anaerobic respiration, but ultimately that process is so inefficient compared to this that not enough ATP will be created to sustain life. The Krebs cycle will also stop because there is no NAD+ or FAD+ being released from the electron transport chain. NADH and FADH2 will no longer be able to drop off their electrons and be oxidized themselves. So the reason we need oxygen is to pick up those electrons as the final electron acceptor and form water. That enables the electron transport chain to flow, and oxidative phosphorylation to produce the large amount of ATP that we need. Oxidative phosphorylation and the electron transport chain provide the body with a gradual release of energy. Because the release of energy is more gradual, it can be more useful to organisms, as they can gain more of that energy and use it in various processes. I hope you found this video helpful. Make sure you know these processes well and understand the importance of each element.